from 2023. It starts with that multiple choice question. They gave us metaphase 1 during meiosis, and they are asking me one simple question here. Look what they said. They said, like, what organelle is this one? Now, the organelle's name, as you probably know, it's the centriole. So you have one centriole and one centriole here, and they're trying to align the chromosomes during metaphase. Next, why is it possible for chromatids to be genetically different from chromatid B? So look at chromatid A, which is this one here, and chromatid B. What's the difference? A is non-recombinant, meaning it's staying as it is. You notice A here on the left? There was no crossing over or recombination of allele happening. But on the other hand, B is actually going through that with another chromosome. Now, here I want you to mind the wording when you talk about crossing over or recombination. Just be aware of the way we write the term. So look what I'm going to say. I'm going to say here, due to crossing over, look what I'm going to say. I'm going to say between the non-sister chromatids. What does that mean? It means that is occurring between B on one hand and D from the other chromosome. Usually you have one chromosome from the mom, maternal, and one from the dad. The crossing over is going to happen between a sister chromatid and a sister chromatid from one of each of those parents. And this results in what? This results in the recombination. Here you must say alleles. And this will lead to what? This will rearrange the alleles. So you get new combination, you get new features leading to genetic variation. Okay? Any question? Here they wanted me just to draw 4 haploid cells resulting from the crossing over that took place on the previous page. It's quite super easy. All it takes is, they drew this for me, okay? So I drew A. I'm just gonna draw B right next to it. So B looks like this. I'm just gonna make that centromere in the middle. I'm darkening it all, and I'm gonna leave one white patch, just like the way they have done it here, okay? So that's one. Okay, I'm gonna just put like centromere here. I'm gonna do the opposite, complete opposite. I'm gonna make it light as a whole, centromere. Just gonna make 2 dark patches. Finally, I'm gonna turn to the non-recombinant one, that is chromatid C on the diagram, just like completely dark. Just gonna make it like fully dark, okay? Any question here? I think that question wasn't difficult at all. Let's get to question 2. They want me to do magnification. So what I have here, I have a scale like this, and they want me to find the magnification of this picture. Now, when you've been given a scale like this, all what it takes is to grab your ruler, measure that scale on your ruler. In my case here, for instance, it is 30 millimeter, like this, like 3 centimeter, 30 millimeters, okay? So I'm just gonna record that size here, okay, to start with. And I want to find how many times the image is greater than the actual. The actual was given there in this scale. So I'm going to say the image is 30 millimeters. The actual is 20 micrometers. So I need those two units to match. So I can match them from milli to micro. I'll multiply by 1,000. Okay, do this here. 30,000, right? Micrometers. And I'll just divide by 20. Image over actual like this. Image actual gives me the magnification. Now, next thing, they are talking here about the process of fertilization in plants. Describe how generative nucleus result in the production of an embryo and endosperm in the seed. You know what they're talking about? They're talking about the process of double fertilization in plants. Let me remind you about it just quickly here. See what I have? Pollen grows a pollen tube. The pollen tube, at the tip of it, I have the pollen tube nucleus. You see it? It's right here. That nucleus here is just to release hydrolytic digestive enzymes necessary to penetrate, just like the acrosome in humans. But then the generative nucleus, it's already haploid, right? Every gamete is haploid. This is gonna divide by mitosis. So if I'm dividing a haploid cell by mitosis, it's gonna give me more haploid nuclei. One will fuse with the polar nuclei. That gives me endosperm, which is triploid. One, and one. So one male, two females. And one will just fuse with the egg cell, which is right here. Learn the position of each nuclei. The polar are in the center, and the egg cell is near the opening, near the micropyle. So that's what I'm going to do here. I'm going to say the generative nucleus divides to form two male gametes, two male sperms, whatever you'd like to call them, okay? And then I say double fertilization. What's double fertilization is all about? It's a male and female nucleus fusing. This gives me a zygote diploid. The zygote turns to what? The zygote will turn now into an embryo. The other male nucleus fuses with a polar nuclei. And this gives me endosperm, which provides what? The polar nuclei, the endosperm, I should say, is the food, the edible part. You see that one here? The one we actually eat. The endosperm is to provide nutrients for the development of the embryo. Okay? All good? Let's check the other one here. State what is the definition of linkage. So, what is linkage? Linkage in genetics stands for the fact that sometimes you have alleles are very, very close to one another. And you know, like recombination from the first question, recombination occurs between adjacent alleles. So, as again, in humans, this trait is the eyes and the freckles and the hair all come linked together. There are alleles that are very, very close, difficult to separate. So when recombination happens, these alleles are not likely to separate. So that's what linkage is all about. Alleles on the same chromosome, close enough that they are unlikely to be recombined. They're unlikely to be separated. I also want to remind you about statistical tests. We've done this in Unit 1. I just want to show you what each test stands for. So chi-square is the one that helps us to determine the difference between observed and expected results. Okay? The other one is t-test. T-test is the best when you are to compare two mean values. Okay? So I have a mean, an average, and an average. I can compare to see, is that difference between the two of them significant or not? And if I have XY, I have a correlation between factor A and factor B, then I go for the correlation coefficient. So again, I'll give you examples for chi-square. Chi-square, if I am to see the difference between Punnett square phenotypes, I was expecting by calculation and observed is the real thing, right? When you see them at the end. T-test, if I have the average size of seeds in one plant and the average size of another plant, and I want to see, is the mean size, is the mean difference between those two seeds is significant? T-test. And if I want to see the relationship between the intake of calcium and bone strength, that's a relationship. So I go for the correlation coefficient. Look what I said, observed, expected. So I go here. Okay? Question 3, they gave us primary wall and secondary wall. You know, like the primary wall is the one that's flexible. That's the one that first forms the beginning when the plant cell is born. Secondary is a commitment. Secondary cell walls occur specifically when the plant cell thickens its wall, turning into xylem or sclerenchyma. So what I have here, showing me the primary on the left, secondary on the right, and use information in the diagram to differentiate, state 3 differences. Now, since this said, the diagram makes my life very easy. Look what I can say. I can say that secondary is thick, primary is thin. That's valid. So look, I'm going to say is thicker than the one on the right. So again, I got more layers, it's thicker than primary. This type of cell wall also contains, I'm talking here about secondary again, it contains lignin, xylem, and sclerenchyma are lignified. Okay? So that gives me what? That gives me strength. In addition, it gives me extra support. You may say waterproofing, it's also good. Okay? Due to the presence of lignin, I can also add less flexibility. Primary is flexible, okay? Secondary is less flexible. And pits are present. Pits, they are the little openings in the secondary cell wall. Finally, another advantage is secondary thinking of lignin is that becomes, I'm referring here to waterproofing. Fact that having lignin adds thickness and waterproofing. Okay? So again, I just want to make a quick comparison here between primary, secondary, just for revision, thin, thick, flexible, non-flexible. Lignin present, lignin absent. Pits, say no pits in primary, right? Waterproofing, so all the things related to lignin are only present in secondary, not in primary. Okay? Someone's asking me about plasmodesmata. They're not related here. You don't have plasmodesmata in secondary wall, only if it's primary wall. Plasmodesmata, I can put it for you here. Plasmodesmata. How long? Desmata. Yeah. So this one is only in primary and is a continuation of cytoplasm. It is connection of cytoplasm between two adjacent cells. Only in primary and is necessary for exchange of materials. Okay? Tell me, how many of these lines here, how many of them are actually correct? Check. I have these 3. See how many of them you expected to be correct. So let's start with alpha glucose. We're talking about cellulose. So do we have alpha glucose? Do we have branching? When I say this one here, that's branching, right? The only correct one is the last one, okay? So it's only one statement. So again, cellulose got beta glucose. Cellulose doesn't have any branching. So the last one, the microfibril part, is correct. Explain how secondary thickening contributes to physical properties of sclerenchyma. So why having lignin again is quite helpful for sclerenchyma. So it adds strength, support, and waterproofing. Now, the waterproofing part is not really necessary here. Because, like sclerenchyma doesn't transport water, but sometimes it's necessary. You know what? If you have the bark of it, if you ever seen, I think everyone has seen a tree in his life. So if you have lignin here at the bark of a tree, that is lignin. It just retains the water inside the tree. So the water stays inside, doesn't evaporate. So that's why lignin is necessary. Good. Now look at the picture. I got pits, cell wall with pits. That is xylem. Okay. Describe the function of pits and yeah, just the function of those pits here. Now with pits, that would allow lateral sideway movement of water. Like xylem looks like this, right? If I have some leaf here, that leaf would get the water from a pit on the side. It's not only about water, right? Because xylem carries water and minerals. So I can just say to them, it's water, minerals, movement is sideways. Good. Here I have what's this picture? It's Caribbean, some forest, and they're talking here about bacteria. Those bacteria is giant, like not normal, right? So they said one bacterial cell is 20 millimeters. That's 2 centimeters. You can actually see this bacteria. Quite interesting. Now, we have three domains of life, right? You know that they're prokarya, bacteria. That's one. What are the other two domains of life? The other one is us. We belong to what we call eukarya, and there is archaea. Okay. Calculate how many times this bacterium is bigger than regular bacteria. So we have this one is just 30 nanometers, and this one's 20 millimeters. So again, I need to do quick conversion here between the units. So one of them is 30, 20 millimeters. I'm just going to do from milli to nano. That's a million, right? So I'm going to say 20 million. This becoming 20 times 10 to the power 6. They want the final answer in standard form. So that's why I'm writing this way. And then I'm going to divide this by 300. Okay? So 20 over 300, both in nanometers. This should give you 6.7 times 10 to the power of 4. Okay? So I just divided the size of the bigger bacterium over that smaller one, and I converted them both to be in nanometers. The coming question is very direct. They gave you DNA, ribosome, cell membrane, and they asked you for the function. Could it be more direct? So they said straight these structures, the ones below, how will they help this bacteria? Again, don't get intimidated by words you've never heard before. They said there is some organelle. This organelle's job is to make proteins. What does it contain? It contains DNA, it contains ribosomes, and it contains a membrane. So how is this structure gonna make those proteins? Let's start with DNA. Think about it. What's the role of DNA in protein synthesis? It's not direct. DNA provides the genetic material to, or the genetic code to synthesize that protein. So I'm going to say it here. Genetic information transcribed so we can make messenger until we turn it into protein. Okay, so ribosomes is what? That messenger we transcribed there from DNA is going to join the ribosomes. And what's it going to become? It is going to become translated into a protein. Now, membranes is like, what does it have to do here? Just think of it that way. If I have this organelle like this, okay? And here I have that circular DNA. Here I have those small ribosomes inside the bacteria. What will be the membrane's role in this case? Now, I could just state that membranes are permeable, partially permeable. They will allow the building blocks, the amino acids, to move in. Plus, being fluid, membranes are fluid mosaic, remember? So they would allow also the export of the product, the protein. So as again, I'm just going to talk in general. Tell them membranes are partially permeable. They allow the import, export of materials or exchange of materials. All right. On the next page, they're showing me a graph. There are two bacteria. Now, they're showing me a graph of percentage oxygen versus growth rate. So I got two bacteria. One is A, one is B. Okay? So again, first thing you look at when there's a graph, you see what's the relationship between what I have on the X and what I have on the Y. Let's talk about bacteria A. I'm just gonna make it pink for, that's not pink, that's pink. All right, so what's the relationship I have here? I'll start. As the oxygen concentration increases, the growth rate is actually decreasing. Okay? So probably this bacteria doesn't really like oxygen. Usually we rely on oxygen for aerobic respiration. So probably that bacteria A is anaerobic. It doesn't require any oxygen. Now, what about A? A is like thriving very well with oxygen, surviving. But again, it's not forever. It's like becomes constant. Now, this is easy to understand. The increase is understandable. It's using oxygen for aerobic respiration. But why is it becoming no more? Why is it not increasing like constantly? Just like in any process in biology, there is not only one factor. There's always many factors. So maybe it has all the oxygen necessary, but there's another limiting factor. There's another factor here that is not been well provided. So think of aerobic respiration. You need oxygen, glucose. Oxygen is good. We have plenty of oxygen, but the glucose is not enough. So that is the reason why it's no longer increasing. Now, since they've put those two bacteria in the same graph, since they've been placed in the same graph, then there's also probably some relationship between those bacteria. It is showing me here that bacteria B survives, A disappears. So maybe that bacteria B has outcompeted A. It's becoming a large number and A couldn't survive that competition. Okay, I'm gonna repeat this. Just give me a moment. Why the optimum temperature and water needed for bacterial growth? You tell them temperature is for enzymes, in water is solvent, hydrolysis, any of these things that are related to water. Just be descriptive. Don't write one word. I wrote these because I know the mark scheme, right? You write it in a sentence showing as many points as possible. Okay? What about here? Explain the results of these two types of bacteria on the graph. So I'm gonna repeat what I've just said. I say as the percentage of oxygen increases, A is decreasing, but B is rising. Okay? I think that's everyone can write that one. This doesn't need any knowledge. But why B is like increasing? That's because it's for aerobic. The growth rate of B levels off. I remind you, it went like this, right? That's B becoming steady. I'll tell them there could be some limiting factor. Maybe glucose concentration is becoming limiting. A is not needing oxygen. A is actually disappearing at high concentration, so could be respiring anaerobically. Here it is an alternative answer. You may say that one of them is more adapted, one of them competes better and like outweighs or takes over the entire habitat for itself. Okay? Which tissue involved in transport of magnesium? Once again, it's same questions repeating. Xylem, right? Water and minerals. Transport, right? They say clearly. Here it says, explain why lack of magnesium could result in yellow leaves and less growth. So, quick reminder about the roles of magnesium. Magnesium is necessary for what? For chlorophyll. Chlorophyll is needed for what? Chlorophyll is necessary for capture light energy for photosynthesis. With photosynthesis, I get glucose. Glucose is two in one. Glucose is just like for all living things, glucose is needed for respiration. But in plants, specifically, glucose is the building block. Cellulose, beta glucose, is also necessary for growth. So I'll tell them here it's chlorophyll, photosynthesis, it's needed, lack of it leads to yellow leaves, and that is mean what that means there's less chlorophyll. Okay? That's because the question says explain. So I explain to them why the leaves are turning yellow. And in addition, it is glucose, and I just told you that glucose is necessary for growth. Here comes that 6-mark question. What is this about? It is about adding calcium. So you see those eggs they're adding, they're breaking eggshells and they're providing into the soil of the plants and as a source of calcium. Okay? Let me just return back to the previous question. She's asking me, do we need to mention ATP? I would mention it. If it occurs to your mind, write it, okay? So I could say glucose is necessary for respiration, providing ATP for growth. Okay? So yeah, I was just about to tell you about calcium. So they provide these plants with calcium. And you can see here in the first part, the area of the leaf is increasing more when I have calcium. Extra calcium gives me bigger surface area of the leaf. And that's really helpful for the plant. More surface area, more light is captured. Okay? Now, let's take a look at the data. Did you notice that the data before this part, like till here, it's all overlapping? What does that tell you? See those error bars are doing like this between the two bars. So that tells you that there's no significant difference between having eggshells, calcium, or not. Afterwards, yes, it is becoming more significantly different. So, like, conclusion, the greater the calcium, the greater the eggshells, the taller the plant is, or the bigger the area of the plant is. Okay? How about this? What effect do we have here? As we have time versus the length of the shoot, with eggshells, it's taller, right? But I have an issue here with this graph. I don't see any error bars. So that's a drawback that could make this graph less helpful. So nonetheless, we have increase with the eggshells. Okay? So again, so far we did two graphs. In the exam, I would just take one graph at a time and write about it. But now I'm just going to go over all of them together. Then we write. Here it's about what? Here's about chlorophyll and calcium. You can see here that more calcium, more eggshells, gives me what? Gives me more chlorophyll. And again, it is less significantly different here at the beginning and becomes more significant with time. So let's write about the first part, the first graph. There is a greater increase in the area of the leaf when? That is when I had calcium. Graph 2, the shoot length is greater. The last graph, also the chlorophyll. So clear conclusion of what we've just seen here, right? Now, let's talk more about the first graph. I say there is no significant difference between the area between 10 days till 24. Why? Remember what we just said here? We said there was an overlap. See the overlap? There's an overlap of these error bars. Okay? The difference was more significant afterwards. Now, the question here, they said discuss benefits of these actual use information and your knowledge. So I need to discuss it. Why is it that calcium helps plants to grow? Again, it seems that they took the entire exam from just a couple of pages because it's about mineral requirements again. Calcium is calcium pictate. What is calcium pictate? Calcium pictate is the foundation of the cell wall. It's necessary for making the middle lamella. Again, the middle lamella is the first layer of the cell wall, and it also forms a network, forms a matrix. This is to strengthen the cell wall, okay? So I'm going to tell them this. Calcium, formation of calcium pictate, the middle lamella, needed to join, to strengthen, providing support for the cell wall. Now, let's just talk because I'm missing a link here, right? You notice I said calcium, cell walls, calcium, bigger leaf area, But what does this have to do with growth? Why is it that calcium gives me more growth? I'll say here that the greater leaf area, the more photosynthesis. So more, just like last question, more glucose. Glucose provides me with what? Provides me with ATP, just like what you were asking, by the process of respiration. And that is needed for what? Why ATP is necessary? It's needed for growth. ATP is also needed for the synthesis of protein. Why am I suddenly talking about proteins here? Listen, it's growth, right? So which biological molecules necessary for growth is nothing but protein, okay? Name a protein needed for growth. You can say enzymes. But in this context, especially because of the last graph, what was the last graph about? What? Chlorophyll. Tell them one necessary protein is chlorophyll. Lastly, I will just like assess the results. Adding eggshells is for free. Adding eggshell is very cost effective. It's not gonna trouble the farmers. And unlike fertilizers, you've probably heard that adding fertilizers cause pollution, eutrophication. Here it is without fertilizer. So it's cost effective and environmentally friendly. Okay? So just to briefly see the format on how we wrote the answer here, it's a discuss. So for every graph, I wrote description, the pro, like what is this graph helping me to do? And I criticize the graph, for instance, about the error bars or lack of error bars, okay? So then I went to explain. So I took the graph, describe, and wrote conclusion, and then I criticized the graph for lack of error bars. And then I wrote explain from the textbook. And finally, I added just a line here just to assess or discuss the advantage of using those eggshells. The next question, question 6, is about some very interesting animal. It's called the mimic octopus. This can turn itself, it's like science fiction, it could turn itself into many things. So it could become like a flat fish, and it could even mimic the shape of a predator. So it has the capacity to turn itself inside out, becoming like a flat fish like this, or becoming looking poisonous, intimidating predators for the sake of what? For survival in his habitat. So again, just think about it. What would be the great advantage of looking like a venomous sea snake? Here, it would help it to being avoided by predator. Predator won't attack a venomous sea snake, right? And here it's trying to look like a flat fish. Here it's trying itself to turn itself into a bait, attracting other fish so I can harm them too. This definition comes like almost in every paper. What's a niche? It's the role of an organism in its habitat. What type of adaptation is what's done here by this octopus? There's only three adaptations we have, right? Anatomical, physiological. What was the third one? Anatomical. Wait, for me, anatomical, physiological, and the third one, it's coming. Someone to save the day. Behavior, I got it. Nobody wrote it for me, I promise. Yeah, behavioral. So What kind of adaptation is this one? When same banding, that's like a color, right? Of a venomous snake. This time they accepted two answers here. They accepted anatomical and physiological. For me, that I would write anatomical all the way, but they've been nice here. They're accepted physiological because it's the making of the pigment is considered as process. Physiology is about process. Anatomy is about visible features. Why this octopus would change its appearance like a flatfish? We said this is to attract other fish so you can hunt them or to just hide. And when it wants to be avoided, it would turn into that venomous snake. Which of the following describe purpose of the current pain relieving drug? What's this about? They've noticed that this octopus here, you can extract some chemical, a drug called T, I'm just going to call it T, tetrodotoxin. And they said, which of the following describe the purpose of the current pain relieving drug in stage 2 of drug trials? So just quick reminder about drug trials. You have the preclinical. Here, you just testing toxicity. You have phase 1 when you're checking for side effects. You have phase 2 when you are actually trying to see the effect on a large sample size. Here I'm doing double-blind trials. Phase 3, I do what? Phase 3, I am increasing the sample size, plus I am comparing it to an existing drug. Here it happens that they did in stage 2 of the drug trials, they compared it to a current pain relieving drug. Why in such studies, human studies, we use existing medication? Why don't I just give them like a dummy placebo? That's because it's unethical, right? You can't leave someone in pain just because you want to try the drug. So you must give them something that will relieve their pain. Now, here's this question, many students got it wrong. They mostly chose placebo here, but they said, what's the purpose of this? What's the purpose of the current pain relieving drug? So the purpose is control. We're using it as a control so I can compare the new medication to some existing drug, right? If they said, what's the name? What is the term used for that? You say, well, yeah, it's a placebo. But here, it's the purpose. So you say, well, it's been used as a control. Describe the processes would have occurred before stage 2 and after stage 2. Okay, so I am right here. I am at stage 2. I am phase 2. I'm comparing it with an existing drug. What would happen before and what would happen after? Stage 1, I am using healthy volunteers. And stage 3, after, I'm using a larger sample size of patients and I'm doing double-blind trials. Now, why we do drug trials in the first place? We do drug trials just to ensure that there are no side effects and I am trying to perfect the dose. Do you know what that happens at the very last stage when everything is done and the drug is being prepared? That's when I publish the results. We do peer reviewing to see the whether to release this drug or not. So I'm going to say now, once it's done, I'll publish, release to other scientists so they can peer review my studies. So that's the very last thing. Now, on the next page, they're showing me a graph comparing this drug, the new one, to the existing one. And they're showing me how much pain relieving they do. So again, I have number of patients, duration of pain relief. So which one seems to be a better medication? Is it the new one, T, or the current? Look, with a new medication, there is longer periods of pain relief. That's the best thing, right, about pain relieving. It's to last longer, all right? So if you compare between the two of them, similarity, they both relieve pain. And what's better about the new medication is that its effect is lasting longer. Comment. Comment meaning what? Comment means how you describe first the graph and you come up with at least one conclusion about whether that data you've been given is valid or not. So description, what am I saying? I say like, look, it both relieve pain. Difference, the new drug relieves pain for a longer period of time. Maybe I mentioned how long it lasts. It lasts like 450, 75, right? 60, I think, right? Yeah. The current is short-termed, so T is better, right? T is more effective. Now, let's criticize the data. Now, we have no error bars, right? We have no control variables. There's nothing being mentioned about variables being controlled between the two groups. We have no mention of the sample size. Pain is subjective, right? Like if I tell you I have headache, nobody would know how strong that headache is, right? So pain as it is not something that would can be compared, right? So that's why I'm saying here, I have small sample size because it's phase two, right? Also, no error bars. So it's not reliable. There's nothing being mentioned about control variables. I say you did not control any of these variables. Patients are not been controlling. So we have the both samples of the existing drug and the new drug, and we don't know what kind of patients we have in each. Now, as I said, pain is subjective and everyone has a different threshold of pain, right? I mean here by tolerance, how much pain one can take. So for all of this, because we are failed to control variables, I'll say, well, this is not a valid, okay? So see what I said here? I said not reliable, while I was talking about the sample size. And here I said not valid when when I'm talking about the lack of variable being controlled. Now, the question before the last, question 7, is about the embryonic stages. They gave me this one here, the morula. Morula is like the 16 cell stage. And they said what describes the morula. So again, in the embryo, we have these two stages. One is 16 cell stage. Those are all totipotent, if you still remember. And this is what I call the blastocysts. That's when I have pluripotent stem cells. So what would we describe in the morula? That diagram they gave you is quite misleading because it looks like a blastocyst, although it's a morula. So I would go for this one here, okay? What is the function of the egg yolk? You know, like in the chicken egg, there is the egg white and the egg yolk. What's the point of having that egg yolk in the egg? Egg yolk is just like the endosperm in plants, right? It's food. It's what provides food for the embryo. Okay? So that's what I'm supposed to say here. Again, it's a suggest question, so it's not something you would expect to learn in the text, but I think you just have to base it on your general information about eggs because the egg white is protein, the egg yolk is all cholesterol, lipids, so it's energy storage. Plus, you can relate it to the plant seed we've seen, where we have an embryo and we have endosperm. This is super important, and as I told you in the last class, it comes like almost every paper. Explain how skeletal muscles can contain same genes, but different structure and function. How muscle cells look different despite having the same genes. What do I call this? What's the title of this when genes are turned on and off? The title is differential gene expression. If they ask you how differential gene expression happens, you tell them by epigenetic control. So I'll tell them here, differential gene expression, when genes are turned on or off. How do you turn on genes? You say by epigenetic modification. What does that mean? This epigenetic modification could involve methylation, that's when you turn off the gene, or acetylation, that is when you turn on the gene, when you loosen up the chromatin and making it accessible for RNA polymerase. Okay? These active genes will get transcribed, forms proteins, and as we always say, those proteins will permanently determine structure and function. All right? Here I have a calculation. They want me just to refer to R8. You see where R8 is? That's the one in pink. And they said calculate how many of those cells are in interphase. But they did give me only the mitotic index. Just quick reminder about the cell cycle, which looks like this. Small section of the cell cycle is mitosis, right? The remaining G1, S, G2, they're all known as interphase, right? So if someone tells you that this is the cells during mitosis, then the remaining cells are in interphase. Again, this is a percentage. So if the percentage is that much mitotic cells, then the remaining cells are actually in interphase. Okay? She just wants to copy this. All right? Yeah, so going back here, they want me to calculate how many cells were interphase, but as a number, not as a percentage. So I have the right numbers in a very silly way so that you don't see them. 2000 cells count And percentage of this. So I'm going to say here it's like 5%, right? That's a 5%. So 5 times 2,000 divided by 100. Me, I'm counting here how many cells are in mitosis. So that's 100. 100 out of 2,000. Okay? Now, if I am to find the percentage, I tell them interphase cells is the total minus the cells in mitosis. So how many of these is going to be? This is going to be 1,900. So I'll repeat. I'll tell them here that there's only 5% of cells are in mitosis. The remaining are in interphase. You may just take 95% and multiply it by the 2,000 and you get those cells in interphase. I just did it in a different way. Either way is fine. As long as you get the right answer, it's okay. Next question. Which cancer cells has the highest proportion of mitotic index? When they say cells undergoing nuclear division, that means which kind of cells have the highest rate of mitosis or mitotic index. It's this, right? So I tell them HCA, the first one. Why people think that research with embryonic stem cells is not a good idea? What's the disadvantage of using totipotent embryonic stem cells? When you use these cells, you're killing embryos, right? It involves always cells taken from IVF, and that involves what? That involves killing or destruction of embryos. And many people think that this is unethical. So I'll say that they will object the use of those because it would always involve killing of embryos, and that is unethical. Okay? Now comes the last question. This one. This is the egg cell. W are follicle cells. They're not really necessary in our syllabus. X is cytoplasm. Y is a haploid nucleus. Z is that jelly coating. We call it zona pellucida. Okay? The student did not include structures that contain enzymes. You know, like in the textbook diagram, it looks like this. You have these, we call them the cortical granules, and those are necessary. They contain enzymes that harden that zona pellucida, this one here. So I tell them, you've missed the cortical granules. Then they gave me two types of birds, this one here and this one here, and they're showing us the sperms. Look, the sperm got here, it got two areas, two main areas here, right? The acrosome and the nucleus. And reminder about the acrosome reaction is like this, right? That's when your acrosome does exocytosis, releasing the enzyme. Sorry, the enzyme's name is acrosin, which digests through that zona pellucida. Then you have those cortical granules I was just telling you about. See them in green? They do their own reaction, cortical granule reaction, to prevent polyspermy. Now, to start with, this said how many of these statements are correct? Let's check. First one says the mid piece of the sperm contains many mitochondria for, there's a mistake here, right? For anaerobic. So that's not right. It's aerobic. Mitochondria only does aerobic. The acrosome is membrane bound organelle containing digestive enzymes. This one is good. Circular DNA, you don't have circular DNA in eukaryotes. Only the mitochondria has, right? If this was the sperm has circular DNA, yes, in the mitochondria, but not the nucleus. The nucleus is eukaryotic, only linear DNA. So that's why there's only one statement is correct. Now, just like the question we did at the beginning, we need to use the ruler to calculate the acrosome to nucleus ratio. So with your ruler, you measure the length of the acrosome and the length of the nucleus for this bird. So you do like this. You measure the length. It's hard for me to do it on my screen here. And then you measure the nucleus. And you divide both numbers by the smaller one. So you get the simplest ratio. Think it's not that difficult, right? So just measure this length, and find the simplest ratio. In the mark scheme, it's that much, okay? Any number between 4.3 to 4.7 and to 1, where the 4.7 is for the acrosome. Okay? Then they asked me to explain how these adaptations related to reproductive behavior of these birds. They said one of the birds, okay, has the large number of the helical membrane inside its acrosomes. Again, the effect of width of the helical membrane on the mean swimming speed of the sperms has been investigated. Do you know what they mean by the helical membrane? They mean this part, where the acrosome. Okay? What do I have here inside? What we have, this area is the one necessary to digest the jelly coating. So the question here says, increase in the width versus swimming speed. So what do we see here? We see as that helical membrane is wider, the swimming speed is more. The acrosome to nucleus ratio and the width of that membrane affect the speed. Why does acrosome affect that? Because it is supposed to digest through the jelly coating. So use information and the graph to support. So I'd say as the width from the graph, as the width is increasing, the speed is increasing. This bird requires a sperm with high speed. Why is this necessary? It requires it because to compete with other males. So that we can have what? We can have higher chance of fertilization. Let me explain to you what I mean here by compete with other males. Just a moment. Back here, do you see what it says? This has several males fertilizers. So the female of the bird mates with several males. So each male, to pass the advantageous alleles for survival, each male tries to make his sperm as fast as possible. And the sperm that comes with the highest speed will reach first. Why large acrosome is good? Why having a large acrosome is good? Because this will get more enzymes to digest the zona pellucida during what I call this reaction again, the one where you're digesting that zona pellucida, we call the acrosome reaction. All right. Here's another thing. I have individual organism produce both egg cells and sperm. They call it hermaphrodite. We've solved another question similar to this. We have this organism that has both gametes, the male gametes and the female gametes within the same organism. They call this in the question hermaphrodite. So they said here, suggest reason advantageous for this animal to have both male and female gametes. Think about it. What's the great advantage of one individual has both male and female gametes? Number one, reproduction. Reproduction is always good for the species. Reproduction meaning that the species is not going to go extinct. If this animal is lonely in its environment, it would always produce offspring. Second, it is the fact that we're making gametes. Gamete, meiosis, meiosis, genetic variation. That's another thing. So again, increased genetic variation. Number two, it's prevent extinction. Disadvantage would be the fact that it is from one individual. Just one parent means less genetic variation because all the alleles are coming from one parent. So first one, I'll say it allows for reproduction, increases the chance of survival, you prevent extinction, and increases the chance of genetic variation because of what? Because although it's one parent, it is still considered as sexual reproduction. We still have meiosis, right? If they ask you about disadvantages, you just tell them there's less variation this way because it's one parent. Last question is just like the one we've been practicing the last lesson. It is the Hardy-Weinberg thing. They gave me recessive, just this one, the white cat. How do I know it's recessive? Based on the symbols. And they want me to find the number of cats that are homozygous dominant and those that are heterozygous. They gave me the formula. Like, look, to start with, the best way to start in these questions is to find the homozygous recessive. So I'll say Q squared equals to 16, okay? Q equals square root, it's a 4. P equals 1 minus Q. So P equals, so Q, P is going to be 1 minus Q, so it's going to be a 6, right? I'm assuming here that is the total number, right? So that's going to be 6. Okay? From the total number of individuals. Now, P squared is going to be what then? P squared is going to be 6 times 6. That is 36. So this is the total number of cats from the population who are what? Who are pure dominant, okay? What if I want to find the heterozygous? The heterozygous one is going to be always 2 times P times Q. So 2 times 6 times 4, that is 48. Okay? So again, if you are to find a homozygous, you do P squared. If you are to find a heterozygous, 2PQ. And the starting point is always going to be from those that are pure recessive. Because if they give you the dominant, there's no way you identify it if it's PQ or if it's P squared. Okay? All right. I'm going to end this class. If you have questions, like one of you has written it here, I'll answer. If you don't, then I'll let you go. So let me answer her. Isn't Q4? No, you don't think of it that way. It is the population what I have here, right? So 16. So you need to find the individual Q here. The individual Q is 4, right? And you have to think of it as total of the number of cats. So if I have 100, right? That's 16 over 100. So you're trying to find the frequency here of those white cats. Okay? So you find it as a frequency, and then you find the individual number of the black to the white. What topics come very often for Unit 2? They're asking me to. You study first the protein synthesis, the modification, the, what do you call it, rough ER and Golgi thing. You study the part about mineral requirements, the cell wall, then mitosis in details, fertilization, that's in plants and humans. You do natural selection, which is super important. And moreover, the one about modification, the one about epigenetics, differential gene expression. Okay, I can send you a whole list of them. We'll have one more class on Tuesday. The next class is on Tuesday, and we have one class before the exam.